guys. I'm gonna finish up this chapter and get over the next one, so that one.
Wait, I'm pretty sure
I'm
this
sure
is on.
we'll be able to cover it. We won't. So your exam's gonna be over these 3 units. We'll be moving along. So we have got to cover some ground. We want to finish up with heredity. Keep in mind there's a lot more for heredity than what we covered, what this chapter and book covers. But you'll get into more of that in cell biology. Last time we looked at Punnett squares. We were getting ready to talk about a couple little things here. We'll move along, like I say, quickly. I won't spend as much time on some of the things that we can do, but there's some that should deserve more attention. Epistasis. Epistasis just means simply a gene at one position, one locus, can affect the execution or the expression of another gene at a different spot. So a gene at one location can affect the expression of another gene. It uses Labrador retrievers as an example. If you've ever seen some Labrador retrievers that are steely black colored, almost steel looking, others that are a dull luster color. Whenever you start to get into things like this, it's not as simple as the Punnett squares that we've shown. You will have a gene in every one of these dogs that says, number one, can they even produce a color or not? You will have another gene that determines, okay, what color is it going to be? Another gene will determine how much of the color will be produced, and so on. So there are many genes, and some of these genes are called modifiers and restrictors and so forth. So it's not just as simple as what is their hair color. There are other things that talk about genes that control intensity, where, how much, and so forth. So, and that's what it tells you here. It says one gene determines the pigment, another gene for color or no color. I mean, there's no guarantee that there wouldn't be color produced. So that is called epistasis. Pedigrees are not something I'm going to spend very much time on. They used to be beneficial, but quite honestly now with genetic testing, they're kind of outdated. So I'm not going to spend a lot of time with that.
Why do we still use it for tracking family ancestry?
Not pedigrees. Pedigrees are terrible for that. I know people
Not Tom uses pedigree, do you?
it used to be, if you go back in time, people would adopt children, take them in, give them their name, they were not even closely related.
Oh, yeah.
So that's the fault with it. That used to be all that we had, and people still use it. Now with genetics, you can tell exactly. There are different kinds of disorders that can happen. If it is recessive, remember that both parents have to give you that recessive allele. And these are just a few examples that we will get into. It says recessively inherited disorders will show up, remember, only if the parent are homozygous for that. In other words, only if the individual is homozygous. Each parent has to give you the recessive allele. If you are heterozygote, you are a carrier. In other words, because it's recessive, one allele will not give you the disease, but it enables you to pass it on. Notice, albinism is a recessive condition characterized by a lack of pigmentation. In other words, the gene that there's going to be a gene that either says, yes, you can produce color, or no, you cannot. If you get both alleles that say, no, those are recessive, then those are albinism, or albinos, which is a lack of pigmentation in skin and hair. Another, cystic fibrosis. And notice this, it's the most common lethal genetic disease in the U.S., striking 1 out of every 2,500 people of European descent. The cystic fibrosis allele results in defective or absent chloride transport channels in plasma membrane, leading to a buildup of chloride ions outside of the cell. And because there's no movement, basically it forms mucus plugs in the lungs. And the lungs just stop working because we can't move out the fluids. That's cystic fibrosis. Sickle cell disease infects 1 in 400 African Americans caused by substitution of one single amino acid in the hemoglobin protein. Hemoglobin has 100 nucleotide units. Substitution of, I think it's the first one, they get switched, and it doesn't work. You've got red blood cells formed that are in the sickle shape. Notice in homozygous individual, all hemoglobin is abnormal. Symptoms include physical weakness, pain, organ damage, and even paralysis. Some traits are dominantly inherited, but remember this, and I will ask you these types of questions on the test. Most of the bad diseases are recessive. The reason is, if they were dominant, then they would show up in heterozygotes, and most people would have the disease. But because many of these diseases are bad, if they're dominant, most people don't live long enough to be able to pass along the genes. So if it's a dominant trait, it's usually will be fatal at an early age. Whereas if recessive, you can be a carrier and keep passing the genes on. Not so if it's a dominant. One of these dominant inherited diseases, achondroplasia, a form of dwarfism caused by a rare dominant allele. And notice up through here, if you have, it's a heterozygote right here, this individual, but they are still have the condition because it's dominant. Normal in this condition is recessive. Why it's recessive? Because many times, particularly in the past, this disease is not fatal, but it shows that many times in the past, if you had the dominant version of a particular disease, it would be fatal. This is not, so you see this. So look, this gives you the Punnett square. So if you've got a heterozygote that has the condition, one that doesn't have the condition, 50% of the offspring will have the possibility of having it. 50% will have the possibility of being normal, unaffected. Which ones are going to be born, no one knows. It's pure, remember, random and chance. As to which condition, but it's 50, odds are 50-50 either way. Yeah.
Did you say this is not fatal?
This one particularly is not, no.
Oh, okay.
There are, most of them are. But this is an example. The reason you see people with this condition out there is because it's not fatal.
Okay.
There is another disease, Huntington's disease is fatal. But the reason it's still out there being passed around, even though it's fatal, is it doesn't become fatal to years, usually after 40 years old. It is a disease that shows up late in life. Even though you carry the genetics for it, it doesn't show up until usually after 40. And so that gives time for people to have children and pass on the genes. But it is a degeneration of the nervous system that just starts to break down. And is probably, I wouldn't say this absolutely, but probably an autoimmune type disease where your body just starts to turn on its own tissue. Genetic testing and counseling, we talked about those pedigrees. They used to use pedigrees and do work backwards to find out people carried the disease or not. Now most of these diseases, all you gotta do is take a little DNA test and they can pretty much tell you what version of the gene shows up. So it's much more accurate. them pedigrees, because remember, just because you've got a granddad Bob, that don't mean he was your real granddad. Does it? You've got a real granddad somewhere, but you don't know absolutely sure it's Bob. Why? People die, people get remarried, people get adopted, and there's no guarantee that just because you carry a name, that the guy you found in the history books 200 years ago is related to you. It may show on paper that he is, but maybe somewhere down the line, some of your ancestors were adopted by their family. So you may not be genetically related, you don't know. And it was real bad for that, particularly in the early 1900s and 1800s, because if there was some one that was orphaned out in the community, usually someone would just take them in, they would assume their name and go on, even though they weren't physically related. So you have no guarantees. Genes, you can change your name, you can change your address, you cannot change your genes. So that's why today, for health issues, if doctors suspect something, they can run a little panel through the genes DNA, and they can tell you what people's got. They can actually tell you pretty much you're going to have blue eyes or brown or whatever. So that test, one of the ways is amniocentesis, which is a kind of horrible thing, I think. But they take the whole wound and just take a needle about the size of pencil lead and stick in through the abdomen into the wound and pull out the fluid from the fetus that surrounds the fetus. It's got cells in it of the fetus, and they can use that then to do DNA testing. So, sounds worse, I guess, than it is. But that is, a lot of that has replaced the pedigree. Now, this is the next big thing. This is what I want you to know better than your parents. Transcription and translation. This is how a protein is copied. The gene is copied and turned into a protein. That is called gene expression. In other words, that gene has a code. Whenever that code becomes functional, it's expressed. Also remember, the purpose of DNA is to code for proteins. That's what it codes for. Proteins control everything about us. So, now are these proteins, how does it go from a gene to a protein? That's what we're going to look at. Those two processes are called transcription and translation. Transcription is writing a copy. If we've got DNA in the nucleus of a cell, if we can't tear the DNA up to get to that gene, it makes all the rest non-functional. So what we have to do is to copy it. Remember, this is in the form of DNA, not a chromosome now. Whenever DNA is in its functional state, it's just DNA. When it's getting ready to be divided, then it gets wrapped in chromatin and becomes a chromosome. So when it's functional, it has to be available. So we've got only in the nucleus then just double-stranded DNA. Remember, transcription is the copying. Notice there is so much proteins do. And we keep talking about enzymes, but all proteins are obviously not enzymes. This is talking some history. I'm not worried about you going into that part that says developing story. This is what I want. The basic principles of transcription and translation. This is hardcore biology that everyone who's going to come out and go into medicine or, who knows, even accounting. Maybe you need to know it. But this is basic. Remember, DNA holds the code. RNA, as it says, is the bridge between the genes and executing those codes to make protein. It's done through RNA. If there, and this is not a 100% true statement by no means, but if you were wanting to say, what does DNA do? codes for proteins. What does RNA do? Makes the proteins. Takes the code and makes the protein. Transcription is the making of an RNA gene copy. There are three kinds of RNA, remember. Messenger, transfer, and ribosomal. The gene copy is messenger RNA. Remember it as the messenger is carrying the message. So messenger RNA is the copy of a gene. That copy will be made in the nucleus. The messenger RNA will then leave the nucleus for us to start to make the protein. So transcription basically means making an mRNA copy of a gene. Translation is to read that copy and to turn it into a sequence of amino acids. Translate like you translate from one language to the other. One language here is the DNA code. The second language is the sequence of amino acids. So transcription is writing the genetic code in the form of mRNA. Translation is the making of the protein. As it says, synthesis of a polypeptide. Remember, when a protein is first made, it is called a polypeptide. There will be parts on that protein sequence that do not make sense. They are there for construction of the protein, but it makes it non-functional until those nonsensical parts are removed. Then, once it's removed, it has the function of a protein, and we'll call a protein specifically. So translation is the synthesis of a polypeptide that will eventually become a protein. Remember, ribosomes make protein. There are two kinds. The kind in the cytoplasm called free ribosomes, the kind that's attached to endoplasmic reticulum. The kind that are attached to endoplasmic reticulum will be prepared for shipment outside of the cell. That is why they are on the endoplasmic reticulum and in association with the Golgi body. So mRNA is the copy. Ribosomes are where ribosomal RNA is where the translation takes place. Transfer RNA that you don't see up here, its job is to read the code and to get the proper amino acid to start the peptide sequence with. The only thing I really want you to remember about this is transcription takes place in the nucleus where it's protected. The mRNA transcript leaves the nucleus and goes to the ribosomes. Before this transcription, this making of messenger RNA can occur, it has to have a starting point. That starting point is called a primary transcript. That's the initial RNA part from which you can start to copy the gene. Now here's what happens in a nutshell. Look up here, this is a bacterial cell, this over here is the eukaryote. That's what we want. There's not going to be that much difference. Up and through here, we have DNA in a cell. Transcription is going to start to copy a section, a gene. And then we will get what is called pre-mRNA. Notice, we copy a section and then it's got those little yellow tags on the end. It leaves the nucleus through these openings called nuclear pores, travels to the ribosome, whether it's on ER or whether it's on in free, in the cytoplasm, depends. Remember, free ribosomes make proteins that will stay within the cell, be used by the cell. Ribosomes that are attached to ER will be taken outside of the cell and released out into the blood usually. Remember, we have got 20 amino acids. What DNA codes for is the sequence of which we start to put those amino acids together. Every protein has a different sequence. Nucleotides code for the amino acid sequence. A series of 3 nucleotides will go for a particular amino acid. Those are called either triplets or codons. Now, this picture here, we've got the DNA. Notice, and remember, in RNA, uracil replaces thymine up here. So we have to unzip DNA and then copy a complementary code. Looks like this one. We replaced the U, then we've got the same for all of the others. Replace the T's with U's. A codon is a series, or a triplet, is a series of 3 nucleotides. They code for specific amino acids. If you notice right here, tryptophan, UGG, that's going to translate into ACC. Any of these in through here, just notice there's different series for each one of the amino acids, and we have 20 amino acids. Over here on the right, it will show you this is the DNA, and these are the little sections that can become genes. Also remember on DNA only actively codes for about 1.5% of everything that's in DNA. 1.5% of it actually code for proteins. The other much of what's there, we don't know what it does, but it probably plays some kind of role in the formation of DNA, protection of the gene. We don't know. And a great number of them are copies of the gene. And one of the things when you take cell biology that you will find, it is not only the code that determines what kind of gene you have, it's how many copies of the gene that will determine what the finished product is.
Did you say that 1.5% of the DNA codes for proteins? Okay, thank you.
During transcription, one of the 2 DNA strands called the template strand provides a template for ordering the sequence of the nucleotides in RNA transcript. The template strand is always the same strand for a given gene. During translation, the mRNA, which was copied made from that, mRNA-based triplets called codons are read in the 5' to the 3' direction. Read 5' to the 3' direction. Codons along the mRNA are read by translation machine. From the 5, starts on the 5' end and goes to the 3. Each codon specifies the amino acid 1 of 20 to be placed at the corresponding position along the peptide. Hopefully you've got a good short-term memory. These are the 20 amino acids and all the codes. You don't have to read
First, I thought you were gonna make us put all that on the test.
that. No point when you got charts to show you. Just understand what they are. These shows the nucleotides and what they can, you can make out of them. Two things I do want you to remember. AUG, August. That is a start codon. That's what tells where to begin. And then there are stop codons. And this is on DNA. And that will tell where the gene starts, and then it will tell you where to stop. Actually, it tells the enzymes. Yeah,
So all three, UAA, UAG, and UGA are all stop codons?
Yep.
Okay, so you remember them.
now remember, the DNA has, this transcript has to start somewhere. It starts with an enzyme, RNA polymerase, that means to make. So it makes RNA. That is the enzyme that is going to construct this messenger RNA. There is one site, one code sequence where it will attach to and start called the promoter. Yep.
Start like halfway through the sequence?
No,
No?
these have to start where they're told to start, and that's what this promoter does.
Yeah, but can that codon be in the middle of the whole sequence?
Of a gene? No, shouldn't be. That would be bad mutation. It has to start at the beginning. And that's what these sequences are. You've got one that says, okay, the AUG, this is the start of the gene. And then you'll have a section that will be called the promoter. And that is where the enzyme can attach to and then start. If you try to start in the middle, it has nothing to attach to. Kind of think of it like a train on tracks. You know, wheels got to fit on the track. This polymerase cannot fit just on DNA except where the promoter is. Then it's kind of like a zipper that you get started zipping. If you get it, miss something down the line, then it doesn't work. It gets thrown off track. That's the way these work. So it's got a place called the promoter where it attaches to. And in bacteria, they have terminators. But I won't do any Arnold Schwarzenegger jokes. But we've got stop codons. The actual good part, the coding part, is called the transcriptional unit. Notice up here, this picture tells you more than words. Up here, we have got the promoter. Notice that's a section of the code. That's part of that stuff that don't code for very much, but still useful. That is where the polymerase is going to attach to. Once it gets attached, then it can start, you notice what it's doing here. These two strands represent DNA, the original DNA. Whenever it attaches to that, it enables it to now separate the DNA helix. This is the template strand. So it will start to make, it'll unwind and then start to copy this bottom strand. As it copies it, then we've got the transcript. Now keep in mind, in that nucleus, in the gel, the nucleoplasm, there will be the enzymes needed to do this, plus the nucleotide resources. Okay. It will go down until it copies it, and then there will be the stop codon, and it will come off. Now, remember it said it will be read in a 5 to 3 prime direction. This end will go into the RNA, rRNA, that makes the protein. It will go into it like a little ticker tape going through a machine, and then it will start to put down the amino acids every time it comes to a codon. So there are 3 parts to this I want you to remember of making this transcript. Initiation, elongation, and termination, and you can count on being asked that. What are the 3 parts of making an mRNA transcript? Initiation, elongation, and termination. And you'll find the same thing for making a protein. So there they are. Now, how does this get started? Promoters tell it where to begin. And notice it's usually, it says here, several dozen nucleotide sequences long that make up that promoter. That is the start point. It cannot start until it attaches. There are other things, proteins called transcription factors that mediate the binding of polymerase. In other words, this enzyme goes up, there's other little proteins, transcription factors that actually help it to bind to the DNA to do its job. The completed bunch of these transcription factors and the polymerase are called the initiation complex. When you get the enzyme polymerase bound to the promoter, helped by these transcription factors, that whole thing, the promoter, the enzyme, and the factors are called transcription initiation complex. Notice this name for the promoter. A promoter called a TATA box is what they're called. TATA is crucial in the formation of this. And what it is going to be a series of TAs, 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 TAs. In other words, it's nonsense as far as making a code, but it's what the promoter looks for to attach to. It knows that's where it's supposed to attach. So up here, you can see the TATA boxes. Okay, we've got the TATA boxes that start, that's where the polymerase will come to. Notice up here, these purple ones are the initiation factors, or transcription factors. And then these transcription factors enable the enzyme polymerase to attach to that TATA box and then to start to make the copy. Notice it's making it in a 5' to 3' direction. And the ribosome will read it in the 5' to 3' direction. It will build the protein in the 5' to 3' direction. Notice it untwists the double helix. It says 10 to 20 bases at a time as it's moving. And it can make a transcript that's about 40 nucleotides long per second. That's pretty fast. It can put 40 nucleotides in their proper place per second when it's copied. A gene can be transcribed simultaneously by several RNA polymerases. Nucleotides are added to the 3' end, so it goes from 5 to 3. And this just is a big blow-up picture of what's happening. Out this end comes the transcript of the messenger RNA. Termination. eukaryote, there is going to be a sequence that starts when we reach the end. And that is going to be a series of just A's, adenines, one right after the other. That's called polyadenylation. Poly meaning many, adenines in a sequence. Those are going to be called the poly A tails. And notice it says there are 10 to 35 of them in a sequence. And they're going to be called the poly A tails. This is done by a second polymerase, RNA polymerase. RNA polymerase 2 transcribes the polyadenylation signal sequence. The RNA transcripts 10 to 35 nucleotides past this sequence. Enzymes in the eukaryotic nucleus modify pre-mRNA. Remember, when the first transcript was made, it was called pre-RNA. During RNA processing, both ends of the primary transcript are changed. That was the yellow parts. Also, usually some interior parts of the molecule are cut out and other parts are spliced together. Now, what they're telling you there, once we make that chain, it's not perfect, it's not exact. There'll be other enzymes at work. They will clean up and neaten up the ends where they're supposed to be. There could be some non-coding parts in the middle, like in DNA, and they will take those out. Introns, exons, remember, exons are the good stuff, introns are the throwaway. We don't know what they do, but they will cut out the parts that are not part of the code. Notice here, on the ends they put these caps. One of those caps is the poly A tail. Here, notice, we've got start codon, we've got the segment, that's the stop codon, that is the protein part, coding part right here, this one little piece in the middle. That right there is the code. Over here, you can see those sequences of A's. Poly A tail is on the end. This is the signal saying it's coming to an end, and then it caps off with just a series of A's, which mean nothing. That gives a place where the enzyme can leave that template without messing up its nucleotide sequence. It gives it enough room. So basically, when it gets off the horse, it lands on solid ground. Okay, you know, it's not coming off in the middle of the signal. So on one end, on the 5' end, we've got what is called the 5' cap. That is there primarily for protection of the ends of the nucleotide. And at the end, after we see the signal, poly A signal coming up that's saying we're done copying, and then it makes a tail to cap it off and protect it. On these parts, non-coding regions of DNA, called the introns. We don't really know what they're for. The coding regions are exons. They're called exon X because they will be expressed. And they will be turned into amino acid sequences. RNA splicing removes the introns and joins the exons. A good question will be, why do they do it in segments? I don't know. I don't know that anybody knows why we have exon or introns, but they're there. This shows you a picture of the pre-mRNA. Remember, it is only these red parts that are going to be part of the code. All these other light-colored parts will be cut out. This is in pre-mRNA. It will remove the introns, splice together the coding segment, the exon. Remember, this is the polyadenylation signal right there, and then the poly A tail, 5' cap, and then this part here where it will attach. So only between these two little lines will be the protein. That will code for amino acid sequences. This is how some of this happens. These little things right here are called SNRPs. Small nucleotide RNA particles. Now, we don't really know that much about what they do, but they're a result of mutations. They don't change. You can actually use them in tracing heredity, ancestry, because they will have the same coding and it just doesn't change. So you can transfer from one group of people to another. You can transfer ancient lineages by that. They come together in a little place called spliceosome. They will cut out these unwanted segments and then make the coded protein. Ribozymes, catalytic RNA molecules that function as enzymes that splice RNA. We can have enzymes that cut out the segments that are unwanted. They will recognize a particular code, and it will say cut here. And then there will be another enzyme that comes along and sees the ends and splices those together.
We will have to remember snRNA. Oh,
Remember
so
what?
the snRNA, I'm not sure how do you pronounce it. SNRPs? snRN, yes.
Those are part of the ones that,
Oh, okay, never mind.
that do that, SNRPs.
SNRPs.
Three properties of RNA are enable its action as an enzyme itself. So we've got the RNA transcript. Not only is it a transcript of a protein, but it can act as an enzyme. And you don't have to remember those three things here. Just remember that RNA can act as an enzyme itself. Here's what you need to remember about transcription and translation. We've made the copy up into the nucleus. The nucleus comes out, the mRNA comes out, mRNA comes out as one segment. It will be modified. That segment is called pre-RNA because it's not finished yet. It will be modified. These ends will be capped and the introns taken out. It's only the coding parts there. That will leave the nucleus through the nuclear pores. If it's going to be the protein used within the cell, then it will go to the ribosomes in the cytoplasm. If it is going to be transported out of the cell like insulin is, it will go to to the attached ribosomes. Here, the 5' end enters here. This brown part is another big protein called ribosomal RNA. Notice it is in sections. It's not just one big blob of a protein. The transcript enters, and when it enters, we see this GGC right there. A transfer RNA will get the particular, this is transfer RNA. The transfer RNA will get that particular code for here. Now remember, G's and C's attach, C's and G's attach, C's and G's attach. So this is the mirror image of it. But now, as this goes into this portal, it will attach right here and then add. See, up here is the actual amino acid it's carrying on its end. It will come in and attach to this here, its code. Then it will release its glycine. In this case, the first one came in was like phenylalanine comes in. It will attach that through here. Notice this is the growing peptide chain right here. Then after it releases it, it will go to this side and exit. So it's got a place it comes, enters, drops off its amino acid that is carried, then leaves. Remember that's goes in the 5', 3' direction. So we've got, all of these little green things are the tRNAs that are carrying all these different amino acids. And only the amino acid that it's ready for can attach. Won't let the other ones, it has to meet this code. This part here is called the anticodon. That's the end of transfer RNA that will attach to the messenger transcript. On the other end of the anticodon, the other end of the tRNA, that is where it carries the amino acid.
If it's going from the right to the left, then wouldn't it be 3 prime to 5?
It's reading from the 5' end to the 3. 5' comes in here, so it reads it. And then it adds it. The next one up, it will add there. Then this one will add. So it's adding in the 3' direction. This is a hairpin, they're called. This is what transfer RNA would look like. These are the nucleotide sequences up here, as you can see. This is where it actually attaches to the amino acid that fits this code. This is the part that will match up then with the messenger RNA. You don't have to remember that particular picture there. This is showing you what the transfer looks like. Here's where the amino acid attaches to, and this is the part that will attach to the code. The anticodon, remember, attaches to the mRNA transcript. The rRNA has those 3 sites we mentioned. The P site holds the tRNA, the A site holds the tRNA that carries the next, and so forth. It goes through the same stages. This is the best way of remembering this. The best way, what are the names of these 3 sites? Just, it's real easy to remember. E, B, A. The E site is the exit site. Right there. That's the exit site, so E. P is the site that holds the transfer RNA that has the growing chain. So that's P. And then A is where it enters. It enters in A, gives off its amino acid in P, leaves in E, exit, EPA. And one other thing, energy is used. This is a copy of a good picture of everything that's happening. Now, I will go back over this. Next time, the first thing, we will run back through that sequence real quick again. You didn't get everything. If you have questions, then we'll have plenty of time to answer. So, after we go through that, then we start with the next chapter. I want you to see what that is, because that one is important.